So the last of our non-living disease agents are prions. So at one time, scientists believed that any infectious particle must contain DNA or RNA. That seemed like a pretty reasonable leap of logic, that anything that can be infecting something else must have some sort of background, some sort of something to direct. So DNA or RNA, which we know as the basis of any cell or even acellular organisms like viruses or viroids or virusoids. However, in 1982, Stanley Prusiner, he was studying scrapie, which is a disease in sheep, a degenerative disease, meaning that they degenerate over time and then die because it's fatal. He discovered that disease was caused by a proteinaceous infectious particle. Proteinaceous, based off of protein, protein-based infectious particle, what are now called prions. Because proteins are acellular, of course it's just a protein hanging out there, it's not a cell, and it doesn't contain DNA, it's just a protein, or RNA. Prusiner's findings were originally met with resistance and skepticism. People were like, how could that possibly be? How could an infectious particle be a protein, just a simple protein that's made? However, it was eventually validated, and he won the Nobel Prize in 1997. So what he discovered was that a prion is a misfolded rogue form of a normal protein. So a normal protein would be identified as PRPC, and then these misfolded rogue forms of these normal proteins, a normal protein being PRPC, this rogue prion protein is indicated as PRPSC. Okay, and that'll be important just when we get down here, which may be caused by a genetic mutation or occur spontaneously. Can be infectious, stimulating other endogenous normal proteins to become misfolded and then form plaques. So within our body, just like we can spontaneously form cancerous cells in our body, right? Our body, while it's going through the process of mitosis and even meiosis, it can form a cancerous cell. Generally what happens is at any given time we have many cancerous cells within our bodies, but then our bodies, being intelligent, are able to seek those out, notice that something's wrong with them, and break them down and reuse the parts. Say you're not welcome, you're not a normal living functioning cell, we're going to make more normal living functioning cells, and we're a healthy body. Similarly, we can have these normal proteins, our body's making normal proteins, and then every once in a while we have a protein that's misfolded. Some sort of mutation occurs, maybe something like radiation happens, or some sort of chemical causes a protein to become misfolded. It's not folded correctly. Our body then will notice this, notice that it's misfolded, and then just break it down into its pieces, recycle the pieces, and then we go about our day. However, sometimes this genetic mutation that happens, or the spontaneous misfolding, ends up becoming infectious. And then when it becomes infectious, that's when we have this rogue prion protein. And then this rogue prion protein will then stimulate other normal proteins to then become misfolded. And then as they are interacting with more and more and more proteins, normal proteins that become then more misfolded and more misfolded, then we get these plaques that form. And then these plaques end up being holes. And then these holes are pieces of missing tissue. The cells aren't able to form there. There's just a big gaping hole in whatever tissue it has infected. So today, prions are known to cause various forms of what are called transmissible spongiform encephalopathy, so TCE, in both humans and animals. So TCE is a rare degenerative disorder, so it's quite rare. It doesn't happen all that often. It is degenerative, meaning that the person is going to become worse and worse over time, and they affect the brain and the nervous system. So that's where we have found prions to be at this time. It's misfolded proteins that are infectious, cause other proteins within brain cells and other nervous system cells to become misfolded. So then this accumulation of these rogue proteins causes the brain tissue to become sponge-like. That's why it's spongiform encephalopathy. and then it ends up killing brain cells and then forming holes in the tissue, which then, of course, leads to brain damage because holes in our brain are not good and they're going to cause problems, so leading to brain damage, loss of motor coordination, so we start to miss pieces of the brain that allow us to control body parts, so loss of motor coordination, and then dementia, so it starts to eat away at the brain, basically. So this misfolded protein is going to cause more misfolded proteins that just create gaping holes in the brain, and then it just keeps going and going and going until eventually the brain has lost all function and then dies because it has so many holes in it. So the infected individuals are mentally impaired and then become unable to move or speak as the brain has become more and more holy. So there is no cure. And the disease progresses rapidly, eventually leading to death within a few months or years. Because as you can imagine, if we just keep making more and more holes in our brain, then eventually the body can no longer function, and then the whole thing shuts down. So here's an illustration of that here on the A side here. We have our regular endogenous protein, the PRPC, just a regular protein. And then when it interacts with or comes into contact with one of these rogue proteins, these infectious rogue proteins, a prion protein, then it's going to cause this protein to then misfold. And then what we get are two now proteins that are misfolded, and then we're going to get this accumulation of these misfolded proteins in this area that then is going to be a whole area. And then, these proteins are going to go on, these misfolded ones, the prions, as they're called, the prions are going to go on and then interact with other proteins, and then that continues on and on, creating these gaping holes. B here is showing a piece of tissue. You can actually see these holes in here. So this is brain tissue, nervous tissue, and you can see all of these white spots are holes, whereas this would normally be filled in. You can see that as well down here. So this would be a normal brain. You can see there's a lot of tissue here. It's very nicely colored, bright. But then here, this is someone that has a prion disease, which we'll talk about in a moment, called Creutzfeldt-Jakob disease. And then you can see that their brain it's not as bright because it has holes in it. So it's not as darkly, or it's not showing up as clearly here because it has all the holes and it's spongy, so it's not as clear on the image. So here you can see normal brain tissue. It's pretty even across that piece of tissue. And then you can see this Creutzfeldt-Jakob disease patient. Their brain has a lot of holes in it. So speaking of specific diseases, there are several of them, the TSEs, in humans, including Kuru, fatal familial insomnia, Gerstmann-Straussler-Scheinker disease, and Creutzfeldt-Jakob disease. So the most common one maybe you've heard of is Creutzfeldt-Jakob disease. It's related to the TSEs in animals, including mad cow disease, and scrapie, which is in sheep and goats. And then also we have chronic wasting disease that are in elk and deer. So you should be familiar with these. Kuru is a very common one. Common for the prions, which is very rare, but Kuru. And then Creutzfeldt-Jakob disease was something that we heard about maybe 10 years ago or so as being a problem. And then in animals, we see mad cow disease, which is related to Creutzfeldt-Jakob, and then scrapie in sheep and goats. And then this chronic wasting disease are in more wild animals like elk and deer. So TSEs, how do we get them? They can be transmitted between animals and from animals to humans by eating contaminated meat or animal feed. So this one specifically is why Creutzfeldt-Jakob disease came to the forefront several years ago, is because mad cow disease was something that was of concern. There were some cows that had mad cow disease, which is a TSE in cows, so they had prions in the brain of cows. And as the cows were going through processing, being killed and cut into pieces for hamburger and such, pieces of their brain, inevitably brain and bone and other pieces of cow, will get mixed into things like hamburgers and steaks and things like that. And so then these prions from these cows that had mad cow disease were spread across all sorts of different hamburger and steaks and various other cow products. And then humans were eating these hamburgers and steaks and cow products and getting the prion in their body, and then getting what's called Creutzfeldt-Jakob disease, which is what it's known in humans, and then had these prions multiplying, as you can say, going through this process in the person's brain, making holes and leading to issues, obviously, leading to death and degradation of the body and the brain and dementia and things. So it can be transferred between animals, like for example, the chronic wasting disease will get transferred between different elk or between different deer, or scrapie between sheep and goats, or the mad cow disease between different cows, or from animals to humans when humans eat the meat from an animal. So if somebody can get Creutzfeldt-Jakob disease from eating a deer, for example, that has the chronic wasting disease. So transmission between humans can occur through heredity. So often the case in the GSS and CJD, which GSS is this Gerstmann-Straussler-Scheinker disease, and then Creutzfeldt-Jakob disease, which can be through heredity or by contact with contaminated tissue, for example, during a blood transfusion or an organ transplant. So if somebody has Creutzfeldt-Jakob disease or GSS, then they get an organ transplant, then they would be giving those prions to the person that they gave the organ to or a blood transfusion to. There is, however, no evidence of transmission via casual contact. So if you meet somebody that has Creutzfeldt-Jakob or GSS or Kuru or fatal familial insomnia, then just shaking their hand or being around them, or even if they sneeze on you, that's not an issue. It's coming into contact with the actual prions that are in the brain or in the nervous system and getting those directly in the body. So here we have our list of our TSEs that are in humans and their modes of transmission. So the CJD is the Creutzfeldt-Jakob disease, and then we have our GSS. And then these are the different types of the Creutzfeldt-Jakob disease. So sporadic, which they don't know how you get it, but perhaps just a normal protein, for whatever reason, having a mutation and turning into our rogue prion protein. Variant CJD is if you eat the contaminated cattle products or by bloodborne transmission. Familial CJD is a mutation in the germline, so something that was passed down through the family, a mutation in a specific protein that then becomes that rogue prion protein. And iatrogenic CJD is getting it from contaminated surgical instruments. So as we'll see here in a moment, prions are extremely, extremely difficult to kill. So even going through regular sterilization processes in a hospital oftentimes does not kill prions. So it's very important to know if a person has that, because then all of these surgical instruments, also other things like hormones and blood transfusion, things like that, will contain the prions. And then when the person uses that, or when a person goes into surgery, if that has been used on a person that has prions, then that would be passed along. Kuru is seen by cannibalism, eating infected meat. So a person that has prions then is eaten by another person, and then they get Kuru. And then our Gerstmann-Straussler-Scheinker disease is a mutation that's passed down through the family. So GSS is not really seen by getting it from anybody else. That's just something that comes in the DNA. And then the last one is the fatal familial insomnia, the key there being familial. This is also a mutation in the germline, meaning that that's something that's passed down in the family. So if you're concerned about prions, generally many of them can be avoided. Like this one, you would likely know if you have that already because it is passed down in the family line. Same thing with the GSS. Creutzfeldt-Jakob, you might know if it's passed down in the family line, but you can avoid Kuru and Creutzfeldt-Jakob by 1, not eating other humans, and 2, not eating other animals. So prions are extremely difficult to destroy. That's what I was mentioning earlier. They are resistant to heat, chemicals, and radiation. So again, even through sterilization methods that are used in hospitals, like even ionizing radiation, they are resistant to. So even standard sterilization procedures do not ensure the destruction of these particles. So currently there's no treatment or cure for any of the TSC diseases, and contaminated meats or infected animals must be handled according to federal guidelines to prevent transmission. So when the big mad cow disease thing came out, it was a very serious issue, and they had to track down all of the different types of products, all of the different cow products, and make sure that they were all disposed of properly so that nobody else came in contact with any of those products to be able to pass that prion disease along.